I'm Maitreyi Parath from the Wong Lab.
Hi, everybody. My name is Shivika Shrivastava, and I'm also from the Wang Lab.
Hi, my name is Sariti, and I'm also from the Wang Lab.
Okay.
My name is Vihika, and I'm also from the Wang Lab.
My name is Sophie, and I'm also from the Wang Lab.
My name's Anagha, and I'm also from the Wang Lab.
My name is Vidya. I'm also from the White Lab.
My name is Kush, and I'm also part of the Wang Lab.
All right, so this is our presentation on engineering bispecific antibodies specifically to target acute myeloid leukemia. Let's get started. So as a brief overview of what we'll discuss tonight, we're going to go over some background on our project and explain what bispecific antibodies are, what acute myeloid leukemia is, and details on why we're doing this project, our methods for our research, some preliminary results, our future plans, and our acknowledgements. So starting off with background.
What is acute myeloid leukemia? So acute myeloid leukemia, or AML, is a blood...
Sorry, it's really hard to hear you. Can you turn up your volume a little bit?
Can you hear me if I come closer? Is this better?
Group members, what do you think? Is it loud enough? Yep, go ahead.
Okay. What is acute myeloid leukemia? Acute myeloid leukemia, or AML, is a blood and bone marrow cancer that is most commonly diagnosed in adult patients. AML can develop from myeloid stem cells and myeloid blast cells, and it makes the bone marrow produce abnormal cancerous white blood cells that deprive healthy non-cancerous blood cells of nutrition. Because AML is a blood cancer, it metastasizes and progresses quickly, using the bloodstream as a method of transport to easily infect nearby nervous and lymphatic tissue, and this contributes to its low survival rate. The 5-year survival rate for adults is just under 30%. Its diagnosis is relatively simple compared to other types of cancer, as it requires only a blood test, which is both non-invasive and low cost.
So what are antibodies? As you can see with the image on the right, antibodies are Y-shaped proteins. They consist of 2 heavy chains and 2 light chains. There is a FC region, which is the constant region that binds to accessory cells and is constant across all antibodies produced in the body. There's also fragment antibody binding regions that are variable and target specific antigens on other cells, shown in orange in this image on the right. These FAB regions are what we are engineering in the lab.
So how does the bispecific antibody work in our project? The bispecific antibody is unique because it has 2 fragment antibody binding regions called FABs instead of 1. For AML in particular, one arm binds to the tumor antigen protein CLL1 or CD123. These are cytokine receptors that are highly and differentially expressed in AML blasts compared to cells typically found in the blood and bone marrow. The other arm of the bispecific antibody binds to T-cell engager CD3. This engagement is crucial as it activates the T cells, which are immune cells responsible for killing cancerous and mutated cells in the body. The end result of the bispecific antibody process is simple: the eradication of the tumor through T-cell proliferation, signaling, and the redirection of cytotoxic response.
There are several risk factors and causes for AML. These include age, as your risk for developing it increases as you grow older, gender, as AML is slightly more common in males than females, exposure to radiation, certain chemicals, and chemotherapy, certain genetic mutations, blood disorders such as myelodysplasia and myelofibrosis, and family history, which is rarely the cause, as inherited genetic mutations are uncommon in most AML cases.
Current treatments for AML include chemotherapy, which is the most common treatment. However, it is non-specific, which means it kills both healthy self cells as well as cancerous cells. Next, we have FLT3 inhibitor drugs that only target variants of AML with mutations in the FLT3 gene, which plays a role in the cell growth process. This is a lot more targeted than chemotherapy. Next, we have IDH inhibitors, which only target AML variants that have IDH1 or IDH2 mutants, which affect blood cell maturation.
The IDH inhibitors block mutant IDH enzyme activity and restore normal blood cell maturation processes. Next, we have the anti-CD33 ADC. This is used in limited cases. The antibody, which is conjugated with the chemotherapy drug, directly binds to the CD33 proteins, which are mostly present on the surface of leukemia cells. Next, we have CAR T-cell therapy. This is currently in development. This involves genetically modifying a patient's T cells, which are a type of immune cell, to target acute myeloid leukemia cells. Finally, we have a lot of bispecific antibodies being developed, and that's exactly what we're doing today.
So bispecific versus the other therapies that are used. So bispecific is better because it trains immune systems to directly produce antibodies. The effects extend beyond when cancer drugs are in the body, and the body itself is trained to fight off cancer. It only kills the harmful cells and none of the beneficial cells, and the side effects generally aren't as harsh. Other methods that are used are radiation therapy, which may damage DNA and organelles, and the costs are extremely high. Surgery, which is invasive, expensive, and high risk, and chemotherapy, which most of you all are familiar with, which uses large quantities of drugs to kill cancer. And like Shivika said, the effects it kills both cancers and beneficial cells.
In this section, we'll be describing and explaining the several methods involved in our experiment. Here's an overview of the experiment. The procedure consists of several steps, starting with the production of plasmids encoding the heavy and light chains from E. coli cells, and then culturing them in agar plates, finally extracting the DNA through MIDI prep. Next, we insert these plasmid DNAs into previously cultured mammalian cells to produce the antibody proteins. After transfection, we extract and measure the protein expression levels to ensure sufficient production, then combine the extracted chains to create functional bispecific antibodies. In the end, we test their viability with MTT assays and check their cytotoxicity. This is in order to ensure the success in targeting acute myeloid leukemia cells.
So the cell lines that we used are HL60, which are the human Caucasian promyelocytic leukemia cell line, and the THP1, which is the monocyte cell line isolated from the peripheral blood.
So our first step in the experiment is to produce plasmids or circular DNA molecules found in bacteria that contain the genes encoded for bispecific antibody variants. This can be done by inoculating E. coli bacteria with our plasmids of interest containing an ampicillin resistance gene. These engineered bacteria contain genes specific to antibodies' heavy and light chains with multiple variants of each component to ensure the best possible outcome for experiment. Overnight, the bacteria multiplies in an agar plate with ampicillin, creating copies of our plasmid of interest. Since only the bacteria that contains the ampicillin resistance gene could have survived, we can be confident that all the remaining bacteria successfully contain the plasmids that we need. Finally, the DNA is extracted through a process called MIDI prep, where bacterial cells are lysed and the cell membrane is disrupted in order to allow cellular components to be collected and non-plasmid cell components to be filtered out. We can check the concentrations of our final plasmid DNA using a nanodrop spectrophotometer.
The next step is cell transfection. After plasmid production, we continue with this step. The purpose of cell transfection is to insert the plasmid DNA into mammalian cells. To do this, one must prepare a PEI DNA mixture and add it to around 50 milliliters of cell culture, and the cell culture should be confluent to about 3 million cells per milliliter. The PEI and DNA form a positively charged complex. This positive charge is what allows the complex to interact with the negatively charged surface of the cell and enter the cell via endocytosis. As a result, the mammalian cells can take in this complex and produce antibody proteins. Approximately 24 hours later, valproic acid, or VPA, is added. VPA inhibits deacetylase, which is an enzyme that decreases protein expression. Thus, adding VPA allows the enhancement of protein expression. The diagram on the right shows a summary of the entire process.
We used flow cytometry to verify the presence of antibodies in the sample by checking for their size and complexity through fluorescence or the excitation of probes. This technique was then employed to analyze the binding ability of these antibodies to leukemia cells specifically using the HL60 and the THP1 cell lines?
So in the final step, we utilize the MTT assay to rigorously evaluate the cytotoxicity and efficacy of our antibody constructs. There are many types of assays such as like MTT, ATP luminescence, MTS, etc. However, we utilize the MTT assay process. The MTT assay is a widely used method for assessing cell viability and proliferation. It evaluates the cytotoxicity and efficacy of antibodies involving the addition of MTT reagent, a yellow tetraxylium salt, to cultured cells. Viable cells with active metabolism reduce the MTT to a purple formazine product. This reduction process is then carried out by mitochondrial enzymes and is directly proportional to the number of living cells. The intensity of the resulting purple color, measured at an absorbance wavelength using a spectrophotometer, correlates with the quantity of formazine produced. A deeper purple color indicates higher cellular metabolic activity and thus a greater number of viable cells. In contrast, a lighter or less intense purple color signifies that there's reduced cellular activity and lower cell viability. As you can see in the picture, both the lighter and deeper purple are both indicated.
Now we'll move on to our results. We produce our bispecific antibodies using VHH1, VHH2, and the VHH3 antibodies. These can all bind to the human CLL1, cynomonkey CLL1, and mouse CLL1 antibodies. The cynomonkey CLL1 is especially important because the antibody must be tested in non-human primates for initial cytotoxicity. And these are all stable at 37 degrees Celsius for 1 week. In the bottom right, you can see our peaks from flow cytometry. They show the binding affinities for different antibodies to our leukemia cells. The secondary antibody by itself, which is a fluorophore, displayed residual binding. CD3 by itself displayed very minimal binding, while all three bispecific antibodies displayed very high levels of binding. This serves as a proof of concept for the viability of our bispecific antibodies.
We then used the results of our flow cytometry and flow cytometry and cytotoxicity analysis to verify that our bispecific antibodies could kill CLL1 positive AML cells. In both our HL60 and THP1 cell lines, we noted that our bispecific antibodies, labeled here as anti-CLL1 CD3, had a much higher cell death percentage compared to mere anti-CD3 antibodies, which are the non-bispecific antibodies. This was corroborated by our flow cytometry results. As you can see, we have the secondary antibody on its own, which shows some binding, and then a similar low level of binding in our anti-CD3 normal antibodies compared to very strong binding of our bispecific antibodies or anti-CLL1 antibodies here. There is a much stronger binding to HL60 as well for our bispecific antibody, and that is something that we'd like to explore in the future. Moving on to our future steps, we'd like to test the efficacy of our antibodies through in vivo assays for AML binding to cells and ensure that the cytotoxicity applies to in vivo assays as well. And also in future, apply the same principle of research for ovarian cancer, going through the entire process of bispecific antibody generation, flow cytometry, and MTT assays, and of course, in vivo analysis.
Lastly, we'd like to give a few acknowledgements. First, to Dr. John Wang for his continuous guidance, support, and mentorship. Next, to ASDRP for providing us with the campus and resources in order for us to conduct our research. And lastly, our parents for driving us around and being patient with us. Thank you for listening, and are there any questions?